Let me ask you something. Have you ever wondered how a perfectly developed drug product, approved and marketed, can suddenly face a global recall because of something present in parts per billion? This is exactly what happened with nitrosamines. Today, nitrosamine risk assessment has become one of the most critical responsibilities for pharmaceutical professionals. In this presentation, I will walk you through a structured, practical, and scientific approach to performing nitrosamine risk assessment for your drug product. By the end, you'll understand not just the steps, but the logic behind them, so you can apply this knowledge confidently in real-world situations. To understand why nitrosamine risk assessment is so important, we need to go back to 2018. This was the year when nitrosamines were first detected in valsartan, leading to one of the largest drug recalls in pharmaceutical history. Nitrosamines are classified as genotoxic carcinogens, meaning they have the potential to damage DNA and increase cancer risk over long-term exposure. This discovery completely changed regulatory expectations. Today, global health authorities like US FDA, EMA, and WHO require mandatory nitrosamine risk assessment for all drug substances and drug products. This is not just about compliance. It is about ensuring that every medicine reaching patients is as safe as possible. Every good risk assessment begins with understanding your product completely. Think of yourself as an investigator. You need to collect detailed information about your API, excipients, manufacturing process, and packaging. Because nitrosamines form only when two key components are present: an amine source and a nitrosating agent. If both exist under suitable conditions, nitrosamines can form. You must carefully evaluate the chemical structure of your drug substance, the synthetic route, solvents used, and manufacturing conditions such as pH and temperature. This foundational understanding determines everything that follows. Without this step, the entire risk assessment becomes incomplete. Now let's focus on the heart of your drug product, the API. Your first task is to check whether the API contains secondary or tertiary amines. These are the most common precursors for nitrosamine formation. Next, evaluate the synthetic route. Was sodium nitrite used anywhere? Were solvents like DMF used? These can contribute to nitrosamine formation or carry over impurities. You should also review the drug master file and supplier information. Sometimes, the risk is already present at the API stage and can carry forward into the final product. Identifying this early is essential. Now let's talk about excipients. Many people assume excipients are inert and safe. But in reality, excipients can be hidden contributors to nitrosamine risk. Common excipients like microcrystalline cellulose, lactose, and starch often contain trace levels of nitrites from their manufacturing process. These nitrites can act as nitrosating agents. So, if your API contains amines, nitrosamines can form. This is why supplier qualification becomes critical. Always request nitrite level information from your excipient vendors. Also establish quality agreements to ensure consistent control. This step is essential in real-world pharmaceutical risk management. Even if your API and excipients appear safe, your manufacturing process itself can create risk. Certain process conditions accelerate nitrosamine formation. For example, heat during drying, low pH conditions, and even water quality. Municipal water can contain nitrites, which may contribute to formation. Another important factor is storage. Nitrosamines may not form immediately. They can form slowly over time during the product's shelf life. This is called time-dependent formation. This is why stability testing is essential. It helps identify long-term risks before they affect patients. Now let's examine something many people overlook: packaging. Packaging is not just a passive container. It can actively contribute to nitrosamine risk. Certain packaging materials contain chemical components that can migrate into the drug product. Rubber stoppers, for example, often contain amine accelerators. Blister materials and adhesives can also contribute. This is why extractables and leachables data must be reviewed carefully. Even small migration over time can contribute to risk. Understanding packaging interaction is an important part of complete risk assessment. After evaluating all sources, the next step is risk classification. This helps determine what action is required. If no amines or nitrites are present, the product is classified as low risk. In this case, scientific justification is sufficient. If there is potential presence, the product is medium risk. Here, targeted screening is required. If nitrosamine formation is confirmed or highly likely, the product is high risk. In this case, confirmatory testing becomes mandatory. This classification ensures appropriate control measures are taken. Risk assessment does not end with identifying risk. It ends with controlling risk. Proper documentation is essential. This includes scientific justification, data evaluation, supplier information, and conclusions. You must also implement control strategies. This may include supplier controls, process optimization, or analytical testing. The ultimate goal is patient safety. A structured and well-documented approach ensures regulatory compliance and protects patients. Let's take a moment to reflect. Nitrosamine risk assessment is more than a regulatory requirement. It is a responsibility. By following this structured 7-step process, you ensure your drug product meets global regulatory expectations. More importantly, you ensure the medicine remains safe for patients worldwide. Because at the end of the day, every risk assessment you perform contributes to protecting human life. And that is the true purpose of pharmaceutical science. Thank you